Hi, everyone. My name's Sebastian. I'm the host of Tears and Rain. And on this episode, we have Christine. The objective of this is to dive into her background, how she got into grad school, her thesis, and the very, very human stories behind the thesis. So without further ado, please, Christine, take it away.
Hi, thank you so much for having me today. This podcast has been really nice to listen to. It's been great to hear people's stories. Hopefully, my story won't be too boring. No, I'm kidding. Um, yeah, so how did I go to grad school? Well, it's kind of funny because I didn't really know what I wanted to do exactly. Um, but I only got into one university. And so the university that I got into, I happened to had said that I wanted to major in biochemistry. And all the other schools that I applied to were like liberal arts colleges. I wanted to go do psychology, but I was not prepared for college. I had a bad SAT, but because I was in a large, not the best high school area, I was like in the top 10% of my class, which really does not mean very much. I'm not proud of that or anything, but I lucked out because in Texas, you automatically got into Texas public schools if you were in the top 10% of your class. So
Interesting.
despite doing abysmally on my SAT and not really knowing what I was doing when I was like writing essays and things, I got to go to a really nice college. And so
Okay.
I was majoring in biochemistry, which was also pretty funny because I was not very good at math at the time and probably still now, but it's very
I
simple now,
feel
so
that.
that's okay.
I feel that.
Um, so I did that, and then, at UT, they had this whole thing where it was called the Freshman Research Initiative. And I guess all the STEM majors would get like this little brochure about doing research as an undergrad. And I was like, oh, cool. I love research. Or I like the idea of science because it's always changing. I was hoping I wouldn't get bored easily. Um, I
Yeah.
knew my dad is a scientist, so I am one of those like 25% easier to become a scientist people. But it was nice, seeing scientists grow up. But I also think my obsession with aliens as a child after watching the X-Files at too young of an age
Nice.
contributed. So I just really liked biology. So yeah, so I ended up signing up for this research thing, and yeah, it was called the FRI. And it was a really helpful time for me because it was learning in a very like low stakes kind of environment because you're not in like a um, classic canonical lab. You're with other students and you have a teacher there. And yeah, so everything's like low stakes. You don't, everyone feels like they don't know what they're doing. So you're not the only one. So I think that's a really like optimal environment to learn how to be a scientist because like you don't have to feel like lonely or anything. So I
Yeah.
think that I think that helped a lot.
What kind of
And then,
topics did you
oh
have
yeah,
during
sorry.
that? Yeah.
Yeah, so that was our goal was to delete a gene from the chicken cell line DG40 cells. And we were going to do it by recombinating, um, which I guess you use a special E. coli strain, I think, and then you heat them up to 42 and they can do recombination. I don't know. It's been literally over a decade. So, but we could never get it to work. Classic um, introduction to biology is
Such a
trying to
real
get cloning
introduction.
to
Yeah.
work. But I do remember my very first PCR worked, and I think because of that, I stayed in research.
Nice. Wow.
Um,
The one PCR this could have made or broken as a scientist.
right? Yeah. No, I mean, but I do think it was nice that it worked, but then everything else failed after that. And then I moved on to like during that time, I went to a lab that studied splicing and yeast, and I had the most amazing grad student mentor ever, and she helped me a lot. And I was studying this enzyme called PRP43, and I wrote an honors thesis and it was really bad, but I like to use it now as an example of how far people can come when they learn how to write.
Nice.
Like here's the before and here's the after. But yeah, and then I wanted to go to grad school. I never wanted to go to med school. I have always been afraid of blood and things like that. So but yeah, and then I didn't really know much about what I was doing when I was applying to grad school. So I applied to some and ended up at University of Pittsburgh, and that was an interesting time. I studied yeast transcription and chromatin. We'll get to the thesis in a minute, but it was a 6 year PhD, so it was a bit long, I guess, on today's standards. But you know, I guess now that I'm in this job, everything before now feels like it was the best thing ever.
I would love to
So
dive into that statement after
yeah,
after the thesis for
yeah,
sure.
we can.
Yeah.
Yeah, we can. So looking back, you're grad school, you don't have to look after anybody but yourself. um, in the lab. I mean, people are parents and stuff, but in the lab before you have undergrad mentees. And it's a fun time depending on the situation. Not everybody has a good time. I definitely had very bad times. I had a lot of really unfortunate personal tragedies
Mm.
during my grad career. And that actually took quite a lot out of me. Um, but we persevered. My lab was really supportive, especially our lab manager slash staff scientist, postdoc person who knows everything.
Mm.
She was like she is still great. Her name is Peggy, and she was very helpful. And she would even I remember one time to get blood drawn and I got really sick. She drove me home and it was just like, oh, you're so nice. But
Everyone needs a Peggy. Everyone needs a Peggy
right?
for sure.
Yeah. And she knows everything too. So it's really nice to have that kind of environment. But yeah, there were like ups and downs. I mean, it was in a very well-respected lab too, the art lab. And I'm very lucky to have been trained there. It's known that lab is known to be very rigorous. And so I think the I had very good training. The best phrase I think I remember from my grad career was, or or maybe not phrase because I can't think of the phrase, but like the idea is like as scientists, we're here to find the truth, and it doesn't matter what the truth is. So I remember learning that when I found out I had an artifact in my experiments. Because a protein tag was affecting the
Oh no.
his yeah, and then we didn't find that out until we had an antibody against. I was looking at histone modifications, and we were using a tag system to look at it, and then it turns out the tag caused a lot of issues. So I had to like repeat a whole year's worth of work, and it was very sad.
Ouch.
But resilience. But I remember my advisor, Karen Arndt, saying, as scientists, we're looking for the truth and we're going to find it and we're going to, persevere. And I did that and somehow managed to graduate. It's funny because I remember being a first year and I was like, I can't even imagine passing my qualifying exam. And then I passed it. And then I remember I couldn't even imagine applying for postdoc and interviewing. And then I did that. And like every time I just couldn't imagine doing it. You just end up doing it when you get to that point. So
Wow.
that was one big thing I learned as well.
So for your first year, did you do a rotation or did you
Oh,
already have your
yes.
topic or how did that work?
Yes. Thank you for asking.
All
Um,
right.
yes, we did 3 rotations. I believe they were like 10 weeks long each. I rotated in 2 yeast transcription chromatin labs and then one really cool evolution development fly lab. Dr. Mark Rabay's was a lab. He was awesome. He still is a great person to be able to talk to. And I just really loved histones, so I ended up joining the art lab because my project was going to be on nucleosome amino acids and how they are affecting transcription. And how the nucleosome itself regulates these things was what I was really excited about. And yeah, and so that's how I ended up in the lab I was in. Actually, it's kind of funny because when I was an undergrad, you know how they say, like, oh, reach out to PIs ahead of time and make sure they'll be taking students. Okay, if anyone's listening to this, you do not actually have to do that. Don't worry about it. Just relax, apply to your grad program. But I did it and I emailed Karen and I was like, do you have any spots or whatever? And I ended up in her lab. So that was kind of cool. But yeah. Yeah, the rotations were great. I think it's an important, what's the word? I'm sorry, I forget a lot of
experience, I
words.
guess. Yeah.
Yes, thank you.
No worries. Yeah. The histone topic, that's really specific. Was there any like external guidance that pulled you there, or were you always just like did you just like open up a book and be like histones are cool? Well, let's try that.
Well, kind of actually. I remember in my genetics class, we learned about the histone code, and I got so excited about it back in the day. And then I took an epigenetics class,
Hmm.
and that fed my excitement
Hmm.
and enthusiasm for chromatin and gene regulation. And even when I was in a splicing lab, I was trying to connect splicing and chromatin. I remember this paper came out maybe 2009, and it was like about splicing and how this histone modification on H3 can help regulate splicing. And I remember being so excited about it and trying to like synthesize what I was working on in the
Hmm.
lab with what I would be excited to do in the future. So that's actually kind of how that happened.
Hmm. That's yeah. I'm always curious about that because it's like it seems like it's a little bit of serendipity and like early exposure to it, right? And
Mhm.
so like these are the type of things that I'm trying to kind of catalog, right? With educators,
Yeah.
or just like how broad of a palette do you expose a student
Mhm.
so that they can find an interest, right? Because I mean,
Yeah.
sometimes from what I've understood, people end up in labs because those are the ones that were available, right?
Yes.
And then they end up like eventually they end up liking it, but there's this just like kind of
Mhm.
like you don't get a chance to really try things out, right? It's like a
Yes.
like it's almost like an arranged lab marriage in a way, and we're just like
Yeah.
you roll up
Yeah.
and be like, okay, what are these things, right? um
Right.
and
Yeah.
so when you got into her lab, did you feel like your background prepared you for it, or did you get a lot of on the job training of sorts?
Ah, both, but I will admit to a little bit of a character flaw in myself is that I always liked being I didn't like taking risks and I liked being like in a place where I felt comfortable. And so, because I was very well experienced in yeast molecular biology from my undergrad, I knew that I would be, happy in a yeast transcription chromatin lab. So I felt actually quite prepared and comfortable in that regard, but I learned so much
Hmm.
still. And like, there was quite a bit that yeast genetics, I learned so much about because the lab I was in, before it was mostly a biochemistry lab.
Hmm.
And of course, the chromatin field and gene regulation from the transcription side of things, definitely. There was a lot to prepare to because this area, I know a lot of science areas can be jargony, but the chromatin field is probably one of the worst in my
Really?
experience
Oof.
because there's just so many letters, especially with histone marks, because it's like I study H3K79 monomethylation, but dimethylation is also very important,
Oh
and
wow.
I was studying, yeah.
Like hyper specifics, yeah.
Oh, yes.
Wow.
In fact, I ended up focusing most of my time in grad school on 5 amino acids in the nucleosome.
Really? Wow.
Yeah,
I mean,
yeah.
sure, like there's, infinite questions out there. So yeah, that
Yes.
wow. So when you when did you kind of finalize the topic that you were going to run for your thesis?
It's definitely serendipity came in to play here because I was pitched a project that an undergrad had started in the power of undergrad research,
That's cool.
started in the lab where she actually was a really cool screen idea. They had this genetic interaction where if you had a deletion of this one gene and combined it with a mutation in one of the histone or histone amino acids that could no longer get modified, the cells would die. And so they used that negative interaction, synthetic lethality, to find other histone modification or other histone amino acids that would be required for
Hmm.
placing that modification on
Cool.
the yeah, it was a very clever screen. So I didn't do that part of the screen. I came in afterwards, and my job was to do like the molecular biology part of it, like find out what are each of these amino acids doing. It was actually interesting because they were playing. Actually, I'm very satisfied with how things played out because I found, or I found that some of these residues in the nucleosome, this was in the nucleosome acidic patch, which is actually a very popular area now because pretty much so many of the structures you see out there have proteins bound to the acidic patch. So it's like
Hmm.
a really important binding hub. It's in the center of the nucleosome. It's got a little bit of a cavity so things can bind. And it's like the only real major acidic region because everything's basic to bind the DNA. But it was really satisfying because I found like these mutants or these amino acids that were important for not just placing the one modification, which was H2B lysine 123 monoubiquitination, but also
There comes
other
the jargon,
ones.
right?
Yeah, exactly. So I'm trying to be not too jargony,
You're
but
good.
also other marks too, like H3K4 methylation and H3K79 methylation. And also, I found some that increased the monoubiquitination mark. And what was exciting to me is that over the past decade, there have been numerous structures of the modifying enzymes showing that they interact with the same region of the nucleosome that we found using genetics and old school molecular biology. So that was nice to hear and see over the years that, this was important.
Yeah, yeah, that relevance part is something that's really interesting because like you
Mhm.
you hear a lot of folks who are just like, I'm not doing anything even remotely near what I did for my
Yeah.
thesis. And yeah, it's just, I mean, that's such a unique kind of experience to say like it wasn't just relevant but important, right? um
Yeah.
and so obviously, like, did you know of it, of the importance potential at the time, like what this could
Oh,
entail?
not in the beginning. I was just like, oh, I'm trying to keep track of all the histones and all these, like, what is this thing? Like, trying to, I remember actually having post-it notes on my wall at my apartment, trying to, like, memorize, okay, dot one methylates this residue, and this is what happens. So at the very beginning, I was just like, this is interesting, but what are the large implications? It took me a while to that's actually one of the big things when I was a grad student, but I think it's a common thing you develop, I guess, is like, what is the big picture?
Hmm.
Everyone asks you that when you're a grad student. And I remember getting asked that all the time because you just get so tunnel visioned, so to speak, on what you're doing at the bench. And so it's harder to take a step back. And, as I got older and through my PhD, I got better at thinking about that. And what was really interesting, actually, some of these amino acid sites actually turned out to be common in certain cancers. And so
Mm.
they're actually some of these oncohistone proteins at this region. And so,
And they bind to the
um,
histones at that acidic patch.
yeah, so having mutations there basically disrupts so many things, and in some cases can be associated with cancer.
Mm.
So, yeah.
Wow, that's just like a handful. How big is that patch that people are studying, like roughly in
Um,
terms of like amino
this
acid
is
count?
embarrassing. I don't even remember.
I know
Maybe like
you're fine, but just like a range, like is it like
I'm like, do
50
I have?
residues or
I
like
would say, yeah, like maybe 20 to 50, like making up the whole
yeah.
area. It's shared between H2A and H2B.
Mm. It's like a contact point area.
Yeah, yeah.
Huh.
Um,
Wow. It's
yeah.
wild
I'm
how that
embarrassed. I don't remember.
you're fine. You're fine. No. No, trivia is for textbooks and LLMs. It's fine. But
Yes.
but it's wild to just hear how important and how studied this tiny patch of
Hmm.
this interface between two
Yeah.
proteins. I mean, granted, they do incredible things, but like just that's the specificity that would always mind boggle me, right? And
Right.
and so did you the peers in your lab were they also working on similar things, or is there a lot of disparate projects going on at that time?
I would say the majority of the lab worked on the PAF1 complex, which is known as the polymerase associated factor 1. And it's involved in transcript, its major role is to aid in transcription elongation. And it also helps promote a bunch of histone modifications, one being this H2B monoubiquitination at lysine 123 or 120 in humans. So I was actually kind of the weird one because I was looking at it from the angle of the nucleosome. But at the end of my grad career, I did indeed link it back to the PAF1 complex and found that RTF1, which is a member of the PAF complex, also binds to the nucleosome acidic patch. I did that using in vivo cross-linking and denaturing IPs, which was fun too. So yeah, so we were all interested basically in the connection between transcription elongation and how chromatin is modified.
Hmm.
Um,
Did you
yeah.
did you feel like you were more independent or a little bit more not micromanaged but more managed while you were in your program?
I would say in the beginning,
Hmm.
definitely had a lot of guidance. It was nice because Peggy was in the lab, so she would give a lot of advice on how to like organize your day. And
Oh, that's great.
I was originally had my bench right next to her, and I remember I'll never forget how this because I do this still to this day. She saw me streaking out a bunch of yeast strains and she taught me to one do the same. I don't know if I can explain this right. So you know how when you have a plate and you're like doing the different phases, she suggested that I do like the same phase for all the strains. So like go through all of the first phase for like 30 strains and then go back and do the second one. So you're doing the same thing over and over again, like, and then you change what you're thinking and doing when you start the next phase, if that makes sense.
And
Um,
for
kind
the
of,
folks like
yeah.
listening, the phases,
Yeah,
how would you
sorry.
describe it a little more like is it the actual
Yeah,
action of streaking? Like
yeah,
the
yes. So here's like you take some E. coli
cool.
and or a yeast and you just like spread it on a plate, and
Yeah.
this would be called the first phase.
Yeah.
And then you take another toothpick, for example, and then you
Yeah.
do the second one. You're further diluting the cells
Yeah.
from this, and then you have another one. And then you try to get single colonies by really spreading out the dilution.
Hmm.
And so if you
I
can
didn't know it was
imagine,
called
yeah,
a phase. Yeah, yeah, yeah.
I
That's
don't know,
interesting.
actually. I could have made that up
Yeah, no, that's fine. That's
in
fine.
my mind.
Yeah, I'm just like yeast people phases. What is this? I like. I mean, I've
Yeah,
streaked a bajillion plates, right? But like,
right. I know. I've seen you on Twitter
yeah,
and at or BlueSky.
yeah. But the yeah, I'm just like I have Swiss cheese in my knowledge base of just like maybe there's some jargon that I missed. So I was about to like Google. I'm just like, what is a phase?
I
Uh,
could be totally making it up.
it's
There
fine.
could be like some
I would
Mandela effect.
I would love to carry on that Mandela effect. No problem. Yeah, I'll tell people their phases. Um, so with the yeah, so like those techniques, if do you feel that without the Peggy,
Mhm.
you would have been less prepared, or would you have stumbled upon the same techniques? It just would have been like a little more friction, I guess.
Um,
Because
I
like, how do we
think
mass produce Peggy if we don't have Peggy?
right.
Right?
I know. I know. I think there are definitely some things that you just come up with, right? Like she came up with that idea,
Hmm.
and that approach, and I have, it's interesting. I feel like there's I can't. I can't think of the word either, but like when there's like bats evolve to have wings and birds have wings, but they're not like the same.
Like convergent evolution, I guess. Yeah,
Thank
there we go.
you.
It,
Well,
it, it feels like Jeopardy a little bit.
I'm
Yeah.
so sorry
No, you're good.
to
You're good.
everyone.
This is really fun.
Um,
Yeah.
like it seems like most scientists do the thing where like if they add something to a tube, they move it to another spot after,
Yeah.
like no one really taught me that. I feel like that's just something we all kind of learn or like do. So it's interesting. So I know that there's like a lot of things like that people just do.
Yeah, there's
So
like, I mean, the same thing. Granted, like, I didn't even have the opportunity of like being benchmates with anybody, right? But I
yeah,
still do the
right.
same thing where I'm like, I move the tube in the row,
Mhm.
and then I move the tube
Yeah.
further up when I did the thing, or I move it back
Yeah.
down, or just little
Hmm.
heuristics like that. But like you said, they don't teach that, and it's just
Right.
it's frustrating because like we're all relearning this like oral history sometimes, or just like
Hmm.
it's common human practice to go like, okay, move the pattern, and then that shifts your
Yeah.
perspective because we're dumb and we forget things, right? Like, yeah,
Yes.
yeah. So
I definitely
I'm
do.
like, I'm very jealous of your Peggy
Mhm.
experience, and I'm just trying to
Yeah,
make
I know. I missed her.
so okay, so to get back to the story,
Hmm.
so let's say your second year, right after rotation,
Hmm.
you have your topic. um
Yeah.
how how is that like in that like your
Oh,
engines are starting to spool up and you're planning,
yeah.
yeah, like how far did you
Oh
have to
man.
plan, or what were some experiences there?
My second year was horrible.
Okay.
It was a very dark time.
Oh man.
It was so stressful because I had to TA at the same time as taking classes and studying for my exam. And that was also the year that I found out about that tag issue. Uh, and I was just in a bad mood all the time.
Oh no.
I questioned what I was doing a lot. Like, oh, I don't even like basic science. Why am I here? So I guess I had a teenager emo period in my second year.
That's
But
funny.
but I did have to write that comprehensive exam. So that helped me plan, I guess. I mean, I just had to, yeah, write down the aims based on what I was already doing in the lab to like basically these are your puzzle pieces. You have to now deal with it and put them together because that's all you have right now. So I had like XYZ preliminary data. So I made aims around those. Um, Planning wise, though, it's kind of funny because I had a whole other project that I was supposed to do and I never did it because this histone thing took off and took up all my time. But I did try to outline, or I did outline that and presented it as like I was trying to look for histone marks or histone residues that were required for transcription termination, so stopping the polymerase. Never really got into that, but if a grad student after me, she did a really nice, beautiful job and got a pretty paper in eLife on it. So
Cool.
Elizabeth. Um, but yeah, so I tried, presenting that as well. There's a lot of holes in my memory from that
It's
time.
totally fine. I'm sure a lot of people listening could reminisce with that. Yeah.
Yeah.
Yeah. And so I guess to not like dive into year two because that was
Mhm.
awful, right? Did year
Sorry.
three get better
I
a little
love
bit?
year three. I
Okay,
think
so
year
tell us
three
about
is
that. Tell
like,
us about year three.
yeah, year three is great. And I also, I mean, I know not everyone's the same, but I think in like commonly the third year is like a really good year for grad school in general because you finished, I think, all of your classes. You might have done TAing or you might be about to TA and it's not that big of a deal because you're done with classes. You've passed your qualifying comprehensive exam, whatever people call it. And I feel like grad students, myself included, you just like change after that. Like
Mm,
it's because you just, I feel like there's a sense, like a type of maturity that is gained after spending months reading papers and trying to know everything about your topic,
mhm,
and then going through that process. But then, like in your third year, it's so nice because you feel liberated to just be in the lab.
mm.
And that's like when I got a lot of data that was good. And I was really happy in grad school during my third year. It
Wow.
was good.
So like emo chrysalis and then emerging afterwards. Got it. Yeah.
Yeah.
Is that, and then so you have like this momentum going towards it, right?
Hmm.
So you started getting data now through your fourth year. Is it just like one after another, just like knocking out of the park all the things that you needed
Oh
to
no.
accomplish, or?
Oh, I wish. Actually, one of the senior grad students in the lab when I was a baby grad student, her name is Monsi, and she would tell me, she would always remind me that like if everything worked the first time, we could all get PhDs in 6 months. Um,
Wow.
so she was very right. So yeah, no, there were yeah, it took so long, and um with histone residue or histone mutants, like some of the histones are some of the most conserved proteins. So mutating them, there's so many like off-target effects.
Sure.
And so some of the phenotypes would be like kind of confusing, and we'd have to, yeah, it took some time
Yeah.
figuring things out.
Uh, did you have like the blessing of automation to help you screen, or is it all like toothpicks manual stuff?
It was toothpicks manual, but luckily I didn't have to do the screening because the undergrad, Ali, she did that already. So that was beautiful. I did a ton of Westerns and chromatin immunoprecipitations. Yeah, it was a lot. But it's good because now I can tell everyone I've made every single mistake there
Hmm.
is with Western plots, so I can help them fix their
Nice.
problems.
I might have to like buy you a coffee for some
Oh no,
consultation when
you
I go
don't.
do it, because
I could do it for free.
yeah, because that's westerns are my nightmare. Partly just because it's so expensive, so I rarely have to do it. And then when you
Yeah,
have to do it,
yeah,
it's like high stakes. You have to do it. Like you
yeah,
need the information, but yeah,
yeah.
definitely gonna hit you up on that. Um,
Please do.
yeah. So through experience and failure came wisdom. Wonderful.
Yes,
Um,
yes.
now by your fifth year, is that when you started writing, or is it even more like screening through the jungle?
Um, the summer, my memory is bad. I believe the summer before my fifth year, I had my paper published, which was so exciting. So I think I wrote it in my fourth year, or I wrote it in my fifth year, either fourth or fifth year. Got published. I remember the review process was very annoying. Um,
Was that your
we wrote
first novel?
it was we and we had a lot of comments. I had to do so many more chip
Oh
experiments.
no.
The rebuttal was 14 pages.
14.
But we got it accepted, and I remember being at my aunt's house for 4th of July, and I got the email that it was accepted, and I was so happy, and I cried.
Yeah, that's definitely
um,
worth tears. Yeah. Rain or
yeah,
not, it's worth the tears. Yeah.
right. And I remember during like while my paper was out for review, I started the this other project, the site-specific cross-linking thing. So I ended up putting this non-natural amino acid on like you can make an amber codon, like a stop codon, and you can use that with a engineered tRNA that will, instead of recognizing that stop codon, it will add the non-natural amino acid BPA, which is not the same thing as what's in plastics. Um, and that amino acid can cross-link, make very short cross-links with neighboring amino acids
Oh,
when
that's
you
cool.
expose it to UV. Yeah.
Huh.
And it's covalent, irreversible, so you can actually like put the protein in urea and you can still have that interaction, so you can get really strong co covalent direct interactors. So I did that with the nucleosome at those sites, sent it off for mass spec, got not the best results back. I needed to do it again, but the results that I did get back, I confirmed by Western that it interacted. So there was histone chaperone, SPT16, and then the RTF1 from the PAF complex. I had just screened by Western blot first, like what are some things that could be here? Because you get like this banding pattern that shifts up. So histones are little, and then you have like a bunch of higher bands. So you're like, oh, what could that be? So you just Western it while you wait for mass spec or something. So
Hmm.
that's what I did. It was a very difficult project though, because it was hard to detect the bands. by the antibody that you're interested in. It could be because the epitope is blocked by the crosslink, or it's just a weak interact or not common interaction.
Hmm.
So I remember troubleshooting so many Westerns. I ended up running these gels for like 7 hours.
Wow.
And I used Tris acetate gels because I read somewhere that the pH doesn't change when you run them for a long time.
Hmm.
Because I was like worrying, oh no, is that causing the band to break down? I don't know. It was so annoying. I'm so glad to be done with that paper.
Wow.
Um,
Yeah, I mean, like if they're uncommon interactions, it'd be like a low population amount of those
yeah.
things. Yeah, that's yikes. That actually sounds like a
It
nightmare.
was horrible, actually. I was very depressed during that time as well. I'm
Um,
hearing, I'm like, oh, I loved grad school. And I'm like, oh, I had an emo year. It was depressing.
yeah, no, but that's definitely understand. I mean, like we're human, right? Like
Yeah.
experiencing
Yeah.
those things and just it's because it's like frustration. I think it was like Churchill or something that said
Mhm.
success is defined as moving from failure to failure without loss of enthusiasm. But
Ah, that's perfect. I
but
should
like
write that in the
yeah,
lab.
but like sometimes you can lose enthusiasm because it's like
Yeah,
you just get bludgeoned all the time, right?
absolutely.
Yeah.
Yeah.
And
That's one. Oh, sorry, go
no, no,
on.
please, please. Yeah.
Oh, I was just gonna say, like, I think that's one reason why I think it's important to do like collaborative projects, because with like colleagues in the lab or somewhere else, because then you always have something else cooking, so to speak. And maybe the other thing will be more successful sometimes. Like maybe they can trade places being good and bad, the different projects.
Yeah.
But that's hard to do as a grad student.
Yeah. And
Yeah.
is because I know like lab culture is a really important part. Did you did you have like a moment where you felt like there was a fit or unfit in terms of like where you were and how
Mm,
the lab was operating? Because I know sometimes people have I'm not saying like personality issues, but more of just like the vibe is just a bit off, right, to what they're normally doing. And I
yeah.
know unfortunately like vibe is a really big thing, right? Because it's like
Yeah.
getting through the
It
hard part
is.
with like
Right.
cranky people is hard. Yeah.
Yeah, I mean, I think I was the cranky person toward the end of my grad stuff because of that Western project. But I think overall, I mean, I think it was a pretty good fit. One of the grad students in the lab, he started just 6 months before me, so we were like buddies the whole time.
Nice.
We were friends, like, or we still are. And we would, aliquot confidence cells together, troll the other grad students together, or
Nice.
the undergrads. We had this running joke that he was 45, even though he was not that much older than us, but he was a bit older, and we convinced them that he like knew was born around Vietnam times or something. And it was just like really fun. And we would bribe other grad students on the floor to bring us coffee while we were aliquoting equal confidence cells. We were like, we'll give you some confidence cells if you go get coffee
Nice.
for us.
There's like
So yeah, no, it was fun. It was
like a
good
trade
times.
agreement going on. That's cool.
Yeah.
Yeah. Oh, that's good.
Yeah.
The so you went through a bunch of ups and downs, but
Yes.
by your sixth year, you I'm assuming you're finishing up or starting to write your thesis, right?
Yes.
And so
I wrote it very last minute. So yes, I was starting to think about it.
sure, sure. So was that because I've heard in a previous episode that they were allowed
Mhm.
to just kind of staple papers together in some of their work.
Yeah.
How was yours more like narrative focused? Like, are you trying to tell a
Um,
story or just kind of report? Right.
yeah, so that first paper I had, I definitely just stapled in, which was great. The cross-linking paper, I actually was really bad. I was one of those bad grad students that PIs complain about, that I left before I finished the papers. And so I actually wrote the paper into my thesis. So that was a bit annoying. And um, I mean, I shouldn't complain that much. It's done. And then the introduction, of course, has to be nice and lengthy. And then I had a bunch of appendices with all of my failed experiments and all the experiments that I did with that tag. Um, so that, was there. Then the most fun chapter is like, what should people do next?
Hmm.
And that's when you get to be like, discussion-y and philosophical, I
Hmm.
guess. That was fun. That was more narrative.
Cool. So by the as the ink was drying on that thesis,
Hmm.
did you feel like you became an expert in your topic?
No, I felt nothing.
Okay. 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 So numb to the world.
Sorry.
No, it's fine. It's fine.
Sorry. I
Yeah.
was so numb. I wrote it in 2 weeks. And I remember my grandma died during that
Oh
time
no.
too. So it was really hard. In the 2 weeks, I had to go to her funeral. And I, I just remember this day, I was like rushing against the clock to get my thesis sent to my committee by 3 o'clock. And I wasn't eating. I was still in bed writing. And my significant other, he like brought me a smoothie. He's like, you have to eat something.
Like setting up
Like
an
that's
IV in
basically,
the corner.
yeah, that's basically where I was at when I was writing the
Oh man.
thesis. I
Wow.
don't think I left the house for 1 week.
Wow. That's
So I was ready for COVID.
right. Wow. So wow. That's so
Yeah,
rough.
it was not good.
It,
It was my fault though. I could have started earlier, but
but it
I
it
didn't.
seems from the stories I've heard that like it's always I could have in hindsight, right?
Yeah.
But it's like also you're experiencing and doing all these kind of things. And were you teaching all the way up till your sixth year?
No, thankfully it was only in my second year.
Okay.
So that was good.
Okay. Yeah. Because like, I mean, just balancing one project plus teaching plus
Yeah.
doing all of
Oh
that,
yeah.
and then life happens to you. Yeah. I doubt anybody listening would blame you, and they're probably all going like, "Yeah,
Yeah.
I secretly did it 4
Yeah.
days before it was due." Um,
Yes.
yeah. So because and it's just so much. It's also like so much to write, right? Like I mean, I
It
love reading them
is.
because I love
It
I have like a
is.
library of suffering that I offer for people
Yeah.
to read other people's theses, but the overall experience, do you feel like it prepared you for what you're doing now, like,
Um,
or was it just like
like
a
the
trial by fire, and as soon as you got through there, you can be a person again?
um so no, I mean it forced what did it force me to do? Um, like writing the thesis, you mean? Because
Just
that
the
part
overall
or just the whole
experience, yeah, like the whole six
yeah.
years that you were there, do you felt like it was
Yeah.
training for what you're doing
Yeah,
now? Yeah.
I think so. Actually, I think it helped. I mean, I learned things. The things that were most helpful were like the grant writing class where
Hmm.
you learn how to write those. Um,
Is that a standard class that are offered in
it
programs?
seems to be our program offers one here. Um, though I remember the presentations class, I got a bad grade in it because I was too jargony.
Hmm.
But what was really funny was I won the poster award the same poster that I got a poor grade on.
Hmm. That's weird.
So,
Yeah.
so I don't know if that class helped very much.
Maybe it
Um,
prepared you for like the hypocrisy of reviewers, where like one person's like publish no
yeah,
edits, the other was like, why are you even
yeah.
dealing me like giving me this? Yeah.
It made me stronger.
Sure. No.
Yeah.
So after you submitted, you defended. How was your defense like? Was
Oh,
that
it was
all a
so
blur, or was it
nice.
cool? Yeah.
Oh my gosh, it was so nice. My friends came to town. Like I had two friends who graduated before me and they both came to see, like to be with my or to see my thesis presentation. That was so nice. I was, yeah, very thankful. And my PI's daughter is really good at baking, and so she baked a cake with the nucleosome on it,
Oh,
and
that's
that
cool.
was so sweet. It was a really nice, a really good, like I had like my, I think I like to tell people, I guess that, and I truly believe this, that like the thing that got me through grad school was my friends. So, like the friends in grad school, having that community, we would play like board games together and things like that. And, having them there, it meant a lot. And so that was really nice.
Hmm.
I do remember my laser pointer died though, and I had to change the battery while I was talking. So,
Were you doing like the cool thing where you just like look at them and just change it on the side? And, yeah,
yeah,
nice. Real pro. Real
apparently.
pro. Nice. Um, so that felt more like a celebration and a bit
It
of.
did. Yeah.
Did you feel any like catharsis or any like relief that it's done, or? Or are you still kind
Um,
of numb from the
I
writing and
was I was numb and I was stressed out because I was like, I need to get everything done before I move
mm,
for my postdoc. And um it's yeah, and I just yeah, it felt weird. I guess surreal, I guess. I
hmm
don't think, but I was also like very stressed out because I was like, oh no, I have so much to do because I wasn't done with that paper. And I
mm,
was like really not happy about how things were because I did not want to leave a paper open. Like
sure.
I was worried about it. I was like, oh no, am I going to lose first author? Like, what's going to happen? Who's going to finish my paper?
Yeah.
It was really stressful.
And that like carryover of a project, even though you're technically done, must have been
Oh
like
yeah.
a like you have no time to even catch your breath. Yeah.
Yeah, and then when I started my postdoc, I had to work on the paper still like in the evenings and stuff. So,
Mm, okay.
I mean, I could have been like, well, I'm not in your lab anymore. I'm not going to do it, which is totally legit for, I'm not gonna, but like for me, because I wanted to get a fellowship and all that, I was like, I need that paper. It's always like the hanging over your head. I
Mhm.
need to finish that paper. I need this paper. It's such a headache.
Yeah.
Yeah.
So for your postdoc, tell us a bit about that. How did
Yeah.
that go?
Um, Ups and downs. I also had a lot of bad things happen in my personal life too. So that was hard, really bad, actually. That was really stressful. But the beginning was good. I went to Seattle, which is like, I miss it so much. I love that city and the Pacific Northwest in general. Um, we drove across country with our cats, me and my partner, significant other. And yeah, I started in Toshi Tsukiyama's lab at Fred Hutch. I also really loved working at Fred Hutch. That place, everyone who's worked there, like when I would ask for advice where to go, they would also be like, I love this place. It's so good. And it's really a great place to work.
Hmm. Did
Um,
you meet the
I tell people,
legendary hermit there as well?
yes.
Yeah.
Oh my gosh, he is wonderful. He helped me with my job
Oh,
applications.
that's cool. That's
He's
cool.
a very good person. He's as nice as he is on Twitter
Wow.
as he is in person,
Wow.
but probably even,
That's
yeah,
probably one
he's
of
a
the
great person.
largest modern compliments you can give somebody is that they're the same as they are
Yeah.
online and they're actually nice people. That's
It's
awesome.
amazing how he helps so many people when he's so busy. Like, yeah, it's incredible.
Very cool.
Huge fan.
Very cool. Yeah, no, likewise, likewise. Like
Yeah.
just I like role model status human being. Very,
Yeah.
very cool.
Right. Yeah.
Yeah. Okay.
Yes.
Tell us a bit about your postdoc. So you got into the
Yes.
lab there, and were you carrying
Yeah.
over similar topics or
Yeah, I love chromatin
totally
and
fine? Yep.
transcriptions
No
and
judgment here.
yeah, so I wanted to stay in chromatin and so I went to Toshi's lab. He had done a lot of genomics. He was like one of the first people in the chromatin field to do these high resolution tiling arrays, so back in the day. So he's like, yeah, good. That was a good lab to learn these things in. And I had no idea how to do genomics. I didn't know how to do anything in the terminal, so I really needed to learn that stuff. And he's also really interesting because he like changed completely from what he was doing around like a few years before I started in his lab. So he discovered like ATP dependent chromatin remodeling enzymes. I switched and did all this really great work when he was a postdoc. And but yeah, and he was biochemist and then became like a yeast genetics person and then started studying quiescence in yeast. So how cells stop dividing and then go into this dormant state. And the reason he did that is because of how like different the chromatin is, the structure of it and is in these cells. And it becomes really repressive and compact. Transcription shuts down, all those active histone marks go away. And so his lab was studying that, and I had read the first paper on that from that lab by Jeff McKnight, um, who also, he was a postdoc there, and he also really spearheaded a lot of work there. And yeah, and so so I was really excited about Jeff's paper, and I was like, he was studying how transcription shut off during this quiescence entry. process. And I was kind of like, at the time, I guess I was a little bit like naive. I was like, I want to study how transcription turns back on, like just
Hmm.
kind of in a broad, like, I don't know. Looking back, I'm like, oh, maybe that was a little, but I did it and it was fun. It was really interesting because you can make the cells in this non-divided, highly repressed chromatin state, and they can sit in like that for a very long time. But when you reactivate them by giving them new nutrients, and they can start transcription immediately, I
Hmm.
found. So
Wow.
that pretty much is what I wanted to study, and that's what I did for the majority of my postdoc. And that's what we're thinking about here in this lab now, though we are going a bit beyond yeast and doing some work in stem cells, because there's just some interesting questions that we can ask there as well. But yeast will always stay close to my heart.
Very cool. Very cool. So you tell us about your position now. Yeah. Where are you now?
Yeah, I am at Ohio State University in Columbus, Ohio. Um, it's a very large university. There's a lot of chromatin people here, which is really nice. The chromatin group has been very welcoming, so I'm very glad. Like we get to collaborate or interact with people in Nationwide Children's Hospital as well. So we have like that monthly group here, and it's a night. I'm in the College of Arts and Sciences, so we're undergrad facing, so to speak. So we have 5 undergrads in the lab, and they are all amazing. I know that I might be biased, but they are. They're mostly molecular genetic. I'm in the molecular genetics department. Most of the students are from there, but I have one neuroscience undergrad. So it's been really fun. And then I also have a lab manager, technician who is amazing. Her name is Miranda, and she like keeps the lab functioning. And she's really good at 3D printing.
Cool.
She
Cool.
has fixed so many things in the lab.
Oh, that's awesome.
And it's been amazing. And I have graduate student Evelyn. She's great working on a yeast project too. And then we recently had a postdoc start in September, Kamal, and she is awesome. I joke with my lab members that like I wouldn't be surviving if they weren't here, but I kind of mean it because they are really that good. I A lot of things were really difficult setting up this lab. It was a very hard time. I did not take good care of my mental health transitioning from postdoc to faculty, and having untreated depression was not good. I mean, I was treated, but it was like not, it was treatment resistant. So at that point, it's when I started, and it just got worse and worse. So I'm really thankful for my lab members and their patience and kindness too, because there were a lot of times where it was just really difficult over the summer, for example, and um, more personal hardships. Life is great. But I overcame that, special treatments, and then now things are great. So I'm happy there. Still getting used to bureaucracy though. That
Yeah.
has not been something I often question if I have the temperament for this job.
Yeah, I guess one question, speaking candidly, like one question would
Yeah.
be just like that transition from you are a bench scientist, you're doing research,
Hmm.
now you're the captain of your ship, but suddenly you are an inbox manager, right? Like you
Yeah.
get
Yeah.
so much like I could like administrative red tape. I'm sure that that
Yes.
was brutal, but like, is there have you been getting any like any training or support from fellow like faculty that kind of helps you understand how to
Um,
even do that dance?
um,
You can skip the question. It's fine.
our admins are great. I'm very thankful for our admins. They've been very helpful, and like I said, like the people in the chromatin field have been very helpful here. Um, yeah.
Yeah, that's totally fine. The so well, in terms of like personally, how do you feel about being a new PI? Like, is it equal parts exciting and scary? Is it mostly scary? Like,
Uh, when I first started, I hated it.
Really? Wow.
I was like, why did I pick this job? Because I was so stressed out and I didn't take time very much. I took 1 week off essentially between postdoc and assistant professor.
Oh wow.
I knew nobody here in the city. Um, I first started, I didn't know how to get into my lab. Uh,
Like physically?
except for, yeah, I had to I didn't have the key to my lab, but thank God for Rachel, she's a new PI too, who started with me. Was here a couple months ahead of time, and she was using my office. And so just thank God I ran into her when I was just sitting in the admin office, like I don't know what to do. So we have supported each other a lot. So I'm hugely grateful for her too. Um, but things are a lot better now, I think. Just getting used to, I mean, the bureaucracy is really hard. And I think it's a running joke that OSU is one of the worst bureaucracies. And coming from a place like Fred Hutch, where there was very little red tape, or everything was contracted on the punch out for ordering things,
Mm.
that was really easy. And there were a bunch of like other chromatin people in our division. So like there would be other people like on the same floor that we could ask for like an enzyme or something.
Mm. Can I
So that
borrow a
was,
cup of your core one kind of thing? Yeah,
yeah, yeah,
that's
yeah.
cool.
So it was very different coming here. But as the year went on and I got to know a bit more, it helps, but it's definitely not as glamorous as people make it out to be. Like I know everyone's like, oh my God, I hope I get a faculty position. And I was that way too. I didn't think I would get one, to be honest. But it's definitely rough and it's I don't know if I would do it again this way. Um,
Yeah.
I do love my lab though, so I am very grateful with how it's turning out. But if I didn't know that, my lab would end up being good. I don't know if I would have done it again.
Yeah, so I guess one of the questions I like asking is if you could turn back the
Yeah,
clock, when and what would you tell your
yeah.
younger self?
Ooh, I would tell my younger self that I need to jump out of my comfort zone more. I didn't jump out of my comfort zone until like the end of my postdoc.
Mm.
So when I started learning cell culture in collaboration with Patrick Pattison's lab, they're great too. So jumping out of your comfort zone, I think, is very important. Learning to do new things is very important. Taking care of mental health fully is very important. And I think, mm, I guess being more strategic about writing papers.
Mm.
Um, so
And then
yeah, I think those would be.
for the few who are new PIs that are listening, what advice would you give them as someone who's just starting? Like,
Feel free to email me. I can send you my quotes and stuff. Um, but I think, yeah, no, I think one of the things that, hiring the best people that you can find is very important. I got extremely lucky. Um, and I think honestly, that's been really helpful to be very um conscientious, but also be really open-minded too. So like I wasn't looking for someone who already had tons of research experience when I was hiring my technician. In fact, she has brought so much to the lab. And she has more of like she works with like she was working at an animal shelter and like, giving them shots and taking blood work and stuff. So like
hmm,
not so much molecular or cell work. And she has been immensely huge addition to the lab. So I think people should keep an open mind. And the most important thing I would say that's kept me alive is hire people who are good at the things you're not good at.
hmm.
So Miranda is extremely organized and she uses calendars and stuff. And so she helps me a lot. So yeah,
That's
she's great.
genuine wisdom right there. Yeah, because
Yeah.
sometimes ego can creep up and saying like, I want folks that can complement, but sometimes you do need a supplementation. Sometimes
Oh
there's
yeah.
parts that,
Oh yeah.
wow.
Yeah, and my grad student knows so much more. She has like knows a lot about like RNA sequencing and long read sequencing. She has experience in, and she's very smart too. And then my postdoc. is an expert in stem cells. So like we're a great team, I think. We all complement each other.
Oh,
So I'm
that's
very
great.
happy
Yeah,
about that. Yeah.
that type of teamwork. Did you, to not to go back to the dark
Mhm.
times, but did you
No,
feel
it's fine.
did you feel the same kind of teamwork, or was there more hierarchy in your grad experience?
Um, Definitely more hierarchy, even among the grad students, like the senior grad students versus the baby grad students. And everyone had their own project. So there was like, and that's a very classic way of doing things. And it's, it's good too. I liked having my own project. I didn't have to worry if like, oh no, I messed this up now. Elizabeth's going to be sad or something. But I do prefer teamwork. I think I started working more like with teamwork when I started collaborating and toward the end of my postdoc. And it was really refreshing. I love it. And we're kind of doing that in my lab now. Like, we're like, okay, everyone, we need to get this paper. We need to get these grants. So like let's do this, this, and this. And I like it that way better.
Just like a pep talk before the game type of thing.
Yeah.
That's cool. Very, very
Yeah.
cool. So at the last couple minutes, if there's
Yeah.
anything you'd like to plug or pitch or just words of wisdom, whatever you'd like,
Oh
the floor
yeah.
is yours.
Oh yes. I think my group, my uh other team, I guess, with Fragile Nucleosome, we have this whole online chromatin community where we have a Discord and we can help each other with bioinformatics questions.
That's awesome.
And then we also have a seminar series that's every other week. And if you're interested in speaking in the seminar series, we have every other week a trainee speak and a faculty member speak. So we like to have give both uh, hierarchy or both career path steps, whatever, you know what I mean. An opportunity to talk, and we're always looking for people who are interested in speaking and joining the Discord as well.
Cool. Awesome. I'll make sure to put the links in the description of the video.
Yeah, I'll send them to
Yeah.
you. Thanks.
Awesome. Well, thank you so much, Christine, for your time. This was really, really fun. I enjoyed it so much. I hope folks listening at
Thanks.
home enjoyed it as well. Yeah.